The overall goal of the following experiment is to use microplate-based oxygen consumption technology to assess the function of the electron transport chain, TCA cycle, beta oxidation pathway, and substrate transporters. This is achieved by first preparing the substrate and injection solutions for the assay. The second step is to add the injection solutions into a previously hydrated respirometric assay cartridge to calibrate the oxygen consumption machine. Next, prepare the mitochondrial substrate solutions and load these solutions in the cell culture plate. Spin the cell culture plate to allow mitochondrial adherence to the plate well before loading the plate into the respirometric assay instrument. Results are obtained that show oxygen consumption rates based on mitochondrial respiration for each respirometric assay.
The main advantage of this technique over existing methods, like the Clark electrode, is that microplate-based technologies allow for more efficient and higher throughput acquisition of oxygen consumption with smaller amounts of mitochondria. Generally, individuals new to this method will struggle because of all the moving parts involved in preparing the solutions for the assay.
On the night before the experiment, hydrate a respirometric assay cartridge in 1 milliliter of calibration solution in a non-CO2 incubator at 37 degrees Celsius. On the following day, take out all frozen stalks and thaw them in a 37 degrees Celsius bath or incubator. Prepare all substrate solutions and injection solutions and store them on ice.